Good morning, everyone. Firstly, I just want to say a bit about myself. My name's Leach, and like obviously a lot of people here today, I have a large interest in the ocean. Recently, over the past few years, my interest has kind of filtered more towards human impacts of the sea, and that's the positives and the negatives. So that led me towards this, which is there evidence that plastic pollution affects coastal biology? So basically with this, I want to look at small plastics around various sites in Cornwall and see if they have the potential to affect different marine species on the coastal environment. This is, I feel, quite an important subject because firstly, for me, it interests me, which is very important in a project itself. And secondly, that it's, in the larger scale of things, it's only been recently discovered. It's known as the invisible problem, which unfortunately to a lot of people, if you can't see it, it's not there, which isn't really the case, and it's allowed it to build up. And that just proves quite how invisible it is. This is a photo of, under a microscope, of a £1 coin and a plastic found at Swamppool Beach in Falmouth. So the aims of this investigation are at 3 different locations. I will analyse the concentrations of plastic at each site. Then at each of these locations I will survey the rocky shore to find out what's there and what these plastics have the potential to affect and also get into the food chain. And what I'm doing today is raise awareness for this subject, as not many people know about this, and as I said, it's quite a new, hot topic at the moment. So if people know about it, then that's one of the aims done. The hypothesis for this is filter feeders in areas of plastic pollution are ingesting plastics. So one of the trickier parts to this whole investigation is to discover where to choose as a sample site. And I decided to pick 3 sites. Firstly, we've got site A, which is Swamppool Beach in Falmouth. It's south coast and it's fairly low in exposure. Then there's site B, Porthtowan on the north coast. It's very high in exposure and is also known for being quite bad in plastic pollution. There's site C, just round the corner from Porthtowan, which is also on the north coast. It's lower in exposure than Porthtowan, but it's in between the 2. All of these sites have a rocky shore with various filter feeders there. So the method to collect the sediment, I'm going to be using a systematic sampling at each site. And I'll be sampling along 3 transits, the high, mid and low tide marks. And then I'll be doing that at each side of the beach and directly down the middle. Each point on the beach, which there are 9 in total, 1 kilogram of sediment will be taken. It's difficult to see with the highlighted bits, but each sediment collection will be done after one of the higher tides of the week. This will just make sure, because I don't know what I'm finding. It's a learning curve, this investigation. So I just wanted to ensure that there's going to be something there, and the higher tides will push that in. So once we've got the sediment, we need to try and extract the plastics from it. And this is the part of the investigation which took quite a lot of different playing around with. But my final method I came up with is to distribute 1 kg of sediment in a 12-litre bucket, then add 3 litres of salt water, stir it for 2 minutes, and then allow it to settle for 2 hours. Once it's settled for 2 hours, come back, pour it through a sieve, and at this point I'll be putting them all 9 buckets together and pouring them all through the same sieve. And then I'll put it in a glass beaker with a little bit of water and put it into an oven for 24 hours. This will dry it all out and get rid of a lot of the organic matter. And then I'm ready to examine it either by eye or under a microscope. Then the final part is count each plastic particle, and this has also been a bit of an issue. I wasn't too sure on how to collect my data once I'd got the plastic, and originally I was thinking about weighing and stuff like that, but as silly as it sounds, I didn't realise quite how small a lot of the plastics were going to be. So the easiest way in the end was to count each plastic particle, and although there is a large margin for human error in this way of doing it, I tried to minimise that by doing this 4 times for each location sample. So starting the investigation, there were many different interesting methods I had to try out. This was actually the first one I did, and rounded up anything I could really find, and decided to turn my car into a makeshift science lab. This was the first and the last time I did this way. It didn't really work, and at no point did it feel very accurate. So then I took the method back and tried to rethink it and redesign it to kind of make it feel a little bit more scientific and a bit more professional. I also treated myself to some new buckets, and I came up with the final method which I've been using. This is the exact same, it's just all the sediment and seawater water will get taken back to the lab, which although that is a slight negative point, that I'm having to remove over 9 kg of sand from each location, but for the investigation it was a lot more accurate and it can be done a lot more precisely. So now we've extracted the plastic from the sediment and we've worked out what is in each location. So now I wanted to survey the rocky shore and see what there is there which has potential to be affected by these parts of plastic. And I tried to keep this simple because the main question is there potential and is there stuff there? So what I did is a systematic sampling method, and I sampled along the rocky shore again from high, mid and low tide points. Then I recorded all the species. Although I will say this was a slight negative aspect to my project because I made the decision to focus mainly on a selected species, which was the common mussel. As I said, it was a learning curve, the whole investigation, and I think selecting the common mussel may have made it slightly trickier and slightly trickier to move on from it, just purely because of the particle size that they are likely to ingest. So the results, each site got sampled twice, and then I came up with a mean for each site, and the more exposed beaches had higher concentrations of plastic, and these are the results before they were averaged. So this is quite an important bit of data, the average size. This was a bit of a surprise for the investigation, how much variation there is between size and different site of plastic particles. So as you can see, site A is much smaller plastic particles than site B. And also with this set of data, as I said earlier, everything got put together, and I liked this because it gave me 9 points of a beach, and I felt it gave a real average overall, like an average sample of each beach, and then I can come up with this figure of average amount of plastics per kilogram of sand. And as you can see in site B, which is Porth Towan, that's 4.5 pieces of plastic for 1 kg of sand, which is quite a lot. And if you were to take that sand and put it into a ton of sand from Porth Towan, there'd be 4,500 pieces of plastic in that bag, which is slightly concerning. So the mussels population, the common mussel population of each site, as you can see, site A followed by site C and followed by site B, and that was done by the same way as the other results, by adding them all together and creating an average. This I kept, as I said, I kept simple because the real question is there potential for this to affect? I'm not looking to see exactly how much there is in each spot. I'm just saying, is the species there which can be affected. So these results don't really mean a lot until you kind of put them all together. And this is the part I found quite interesting because I wasn't really expecting it. But if you see as site A has the smallest amount of plastics, but yet it also has the most amount of mussels and the smallest size of plastic. So for the common mussel itself, site A has a large potential of plastics getting into the food chain and affecting the common mussel, purely because of the size. And if you don't mind, I'll read a quick paragraph from my final write-up. At the moment, a hypothetical conclusion can be reached, as the results show there is filter feeders present in areas of plastic pollution. This finding correlates with other investigations, such as one looking into bivalves ingesting plastics. Stephanie L. Wright, 2012. This paper shows the most common size plastics ingested from mussels were 2 to 16 UM, with plastics up to 1 millimeter. This correlates with site A's smallest plastic particle, which was recorded as under 1 millimeter. So although I can't say right now that I have opened up mussels and found plastics, I've used other data which is out there, freely available, and come up with a hypothetical conclusion. So the statistics for the investigation, I was collecting quantitative and qualitative data, and I used one-way analysis of variance statistical test. I used this because there were a lot of means involved, and the plastic concentrations were statistically different, and the filter feeders, no statistical difference. So I definitely wouldn't say this project is over. There's definitely a lot more room to continue with this, and I do hope to continue with this next year. Some of my plans are, I'd like to go to each location and extract the same plastics and take mussels from each of those locations and set it up and try and artificially create and see if they are getting ingested. And I'll do that in a tank. Also, as I mentioned earlier, I feel like I kind of shortened this investigation purely down to time, but I would like to look into other species because the overall question is it affecting coastal biology, and that would include more than just the common mussel. Like all investigations, there are negative aspects to this project. So as I said, I haven't got data, I haven't opened up any mussels and found plastics, but I have managed to use other data available, and I've managed to find the plastics in the area which are small enough for the same species in that area to ingest. Like I mentioned earlier, there is a large margin for human error. That's a lot down to the counting. I would like to change that, but there are some aspects which there's not much you can do. I tried to minimise it by doing it as many times as possible, but there's only so much you can do about that. Also, I would really, if time wasn't an issue, I would have definitely done more sampling at each location. Unfortunately, only 2 sampling sites got done, 2 samples each site got done, and that's purely down to time. So research, as I said, it was a learning curve, so I kind of had to mix and match a lot of different papers and methods and whatnot to figure out how I was going to do this. I didn't really know much about the subject before, but I was really interested in it. So a lot of these papers, I mixed and matched the methods, I've adapted them, I've tested them, and then I've come back and I've finally been able to create the method for me, which has worked.